All right, so I know on Monday we didn't quite get to the end of our lecture, so we're not quite going to move on to DNA replication yet. Right? We ended the lecture talking about chromosomes. I've given you all of these fill-in-the-blank ones on your slides because we already went over these. Right? So remember, chromosome is that discrete unit of genetic material. If we look at our images, I have 2 chromosomes here, and I have 2 chromosomes on the other side. Right? The difference is that on the other side, it's gone through replication. So it has sister chromatids attached at the centromere. Whether it's been replicated or not, we would still call them homologous pairs because they carry the same type of genetic information. Right? You're just getting 1/2 from mom, 1/2 from dad. They are pairs. That's a good question. Yes, so this is a homologous pair. They just haven't been replicated yet. Yep. And this is how chromosomes will look most of the time in our cells. Right? And we'll see when we talk about genetic replication, they don't actually get that X shape until a cell is about to divide. All right, so I left you with this slide. And so I want to give you maybe a minute to talk with your neighbor to review what your answers were, and then we will go over the answers together.
Yeah, 2 is the centromere, and then the 3 is the sister chromosome.
Oh, yeah, yeah. One would be, no, I take that back. One is, one was, 2 is.
I was thinking of you when I saw that. At least a couple.
I got half a point. If I got half a point,
At least
you only get half a point.
There's 3 of us.
It's so bumpy. Yes. No, she definitely should say half. Yeah, so that's 0.25.
Yeah. Yep. The only difference is homologous pairs. Because they carry the same type of. So there's like eye color. Both of them carry eye color. They're just maybe a slightly different variation. One variation you got from one parent, the other variation you got from the other parent. And when we talk about genetics, we end up talking about the dominant. That's the real capital. But there's no carry on those chromosomes. So chromosome can either be a single one or it can be one as an X. The only difference is when it's an X, it's gone through replication. So it's still a single chromosome.
That was my best.
Very nuanced terminology.
Yeah.
And we will go over this. All right, if I were to look at the image and ask you, what is number 1 showing me? What term? What would you tell me? Homologous pairs, yes. They're each individually a chromosome, right? They carry the same information, right? They might carry a slightly different variation, right? So if it's eye color, maybe it's 2 different eye color variations. One's coming from mom, one's coming from dad. But it's the same type of information. What about, I'm going to skip to number 3. What would I say number 3 is? Sister chromatids, yeah. So if we look, sister chromatids, we're looking at 1 chromosome, right? That's in an X shape, which means it's been duplicated, it's been replicated. And either side contains the exact same information, right? So if this one came from mom and mom passed along the gene for blue eyes, right? On both sister chromatids, it's going to be the gene for blue eyes because it's an identical copy. What about number 2?
Twin.
Centromere, yeah, it's kind of the middle of the chromosome, or roughly the middle of the chromosome. Right, it's where the sister chromatids are attached together. Really easy way to know if these are a homologous pair. Right? Something I haven't mentioned quite yet, and we'll come back to when we talk about mitosis and meiosis. But normally they're going to be the same length, right, and have the same shape to them. Right, so if you look at this one, roughly the bands are the same with a little bit of variation on them. So roughly the same shape, roughly the same size. All right. We're going to touch base on this terminology a lot when we talk about cell replication. But the last level of organization for genetic information, the final stage, would be the genome. Right? And the genome refers to the complete set of your genetic information, right? So all the information that a cell needs to make you, right, or do the processes in your body, right? That is the complete set. So up here we have the genome for us, for humans, right? Our complete set consists of 23 pairs of chromosomes. Each pair is homologous. Right, so each pair, we have one from mom, one from dad. Each pair contains the same information, right, but slightly different variations. And if we add all the 23 together, right, that's what makes us you, right? That is your genetic identity, right? So we have 22 chromosomes that we would call autosomes, and then a 23, which is our sex chromosomes, where you're either an XY or an XX, right? So identifying you as male or female in your chromosomes. All right, any questions about our genome or chromosomes? All right, so we now see how DNA is organized. Right, we know we start with our double helix, right? Double helix will be condensed into our chromosomes. All of our chromosomes together are genome. Now we're going to move on to how this DNA is actually replicated, right? So when it's replicated, we get that iconic X shape in our chromosomes, right? And it's going to follow the process that we're going to learn about now. All right, when does replication happen? Replication only happens when a cell is about to divide, right? So if your cell is not about to divide, you're not going to have that classic X-shaped chromosome. You're just going to have this singular chromosome structure, right? A singular chromatid. Now, when a cell gets ready to divide, maybe it's replacing a cell that was damaged, right? Or maybe it's rebuilding some tissues. You fell and scraped your knee, you have to rebuild that tissue, right? A cell is dividing. We have to replicate our DNA. And when DNA is replicated, we get this iconic X shape that will bring through mitosis and end up with two identical cells to the original. Right? Now we're going to focus on mitosis and meiosis on Friday. Today we're just going to focus on replication. So how do we go from a cell that's not yet dividing to a cell that's ready to divide? Right? So how do we duplicate our information? All right, there's some terminology that we have to get out of the way because I am going to use this terminology very frequently. All right, if we look at our strands of DNA, right, we have our double helix here. Right? The strands that are going to be used as a template, or the strands that are going to be kept, right? The original strands, we would call the parent strand. Right? In this image, it's represented as blue strands here. So we have our original DNA. We see that this DNA actually splits, right? And then each resulting DNA has one parent, and then it has one daughter strand. The daughter strand is what's newly built, right? Newly synthesized strand. All right. So we have our parent strands. That's the original. We have the daughter, that's the one that's newly made. Where do we think this happens in the cell? In the nucleus, yeah. DNA cannot leave the nucleus, so this all happens in the nucleus. So going from a parent strand to a daughter strand, making those daughter strands, it all happens in the nucleus. All right, so DNA replication. We didn't always know how DNA was replicated, right? We had to do a lot of experimentation to figure out exactly the method that this was done. So there are some early proposed models of how DNA was replicated, right? So there was 3 of them. There was the semi-conservative, the conservative, and the dispersive. The conservative proposed model states that, and it's the top picture here, we went from our original double helix, we split it in half so that the resulting DNA has 1 strand from the parent, 1 strand from the daughter, right? So we're semi-conserving, conserving half of our DNA. Now the conservative method said, well, we're not going to split that DNA molecule in half. In fact, a lot of scientists didn't think that we could break the hydrogen bonds between our double helix to split it apart. So a lot of scientists thought maybe we are replicating our DNA via this conservative method, where we have a parent, right, our original DNA, making 2 strands, or keeping those 2 strands and making a DNA that's completely daughter strands, right? So we went from a parent strand, replicated that parent strand, made something that was completely a daughter strand, right? We're not splitting that DNA in half. And then we have this last proposed method we call the dispersive method. And this method essentially says we're chopping up our DNA into chunks, we're replicating chunks, and then we're throwing it all back together. So if we look at our parent strand here, right, DNA replication is happening in chunks. We're throwing it all back together so that each resulting strand has chunks of parent, chunks of daughter, chunks of parent. Right? So there are these 3 methods, and we really didn't know what method was the right method, right? And because this next video can draw it on a whiteboard way faster than I could ever draw out a visual, I have a nice video that we're going to watch that helps explain what these methods are, what these theories are, and the experiment behind how we came to our definitive answer. Exam.
Yeah. Thank you so much.
That's okay.
I saw you. Yeah. Thank you.
Yeah.
Thanks so much.
I mean, it made the results difference.
Not necessarily experiments.
Yeah.
One was
All of the content that I was focusing help us understand how we got any methods we don't have.
And then they added in light, and then it showed the and you're still gonna scroll. He says that's this one, what colors? Yours is like, looks like.
It's far away. Oh, it's like far. How far away from you?
He's a JV.
40 minutes. It's not bad. That's an hour. Yeah. Well, like more northern. Yeah, well, yeah. We're in Minneapolis. Okay. And they're coming out. Yeah. I like heard all of it. Like I have so well. Like I'm trying to drive through it.
All right, so in this experiment, they use this heavier nitrogen so that when the DNA would replicate, it would incorporate this heavy nitrogen into its molecules. What was the purpose of it? Why was this heavy nitrogen important in this experiment? What did it tell us? Yeah?
Which strands were like the parent strands compared to which ones were not?
Right. It told us which ones were like the parent strands and what the resulting daughter strands looked like or how heavy they were, right? So basically, we grew DNA in the presence of heavy nitrogen, right? So all of our DNA was heavy. Then, taking that heavy DNA, we incorporated some lighter molecules. So by incorporating the lighter molecules, we would know the newly synthesized strand. Is it all heavy? Is it all light? Is it a mixture of both, right? So by being able to use those different weights of nitrogen and centrifuging it out, right, it's a very simplified experiment, but it led us to determine that that semi-conservative replication method was the method of DNA replication. Any questions about it? All right, well, let's talk about exactly how DNA replicates, right? So we know it has the semi-conservative method of replication. So we know we take a single double helix, split it in half. Each half provides the template to synthesize a new strand, and thereby creating two new double helixes, right? We also know so far that nucleotides in DNA have to obey the rule of base pairing, right? A is always going to pair with T. C is always going to pair with G, right? Never other ways around, right? It's always that rule unless a mistake is happening, which is very rare. We also know, and we'll touch on this a little bit later in the slides here, that DNA has directionality, right? We know one strand is from 5 to 3, the other strand is opposite from 3 to 5. And that's also going to make a difference in how DNA is replicated. Right. So we're starting with one double helix. We're getting two double helix. Right. The original double helix is being split in half. Those hydrogen bonds are splitting to create the template. So let's start at the start. All right. The first thing that happens is we get this origin of replication. So this is a particular sequence in our DNA that signifies that this is where we are going to start unwinding our DNA, right? So this is the origin of replication. Now, the origin of replication is going to be the start of where we unwind our DNA. When we unwind our DNA, we get what's called a replication bubble, right? Because if we look at this image, it looks like there's a bubble inside of our double helix, right? So this bubble is where we are unwinding our strands, we're breaking those hydrogen bonds. Now at either end of the bubble, right, so the bubble has two ends that are connected to double helixes still, we would call those ends the replication fork, right? So that is the site of unwinding. Okay. So notice when we start replication here, we're kind of starting in the middle of a DNA strand. right? And that bubble is going to get bigger, and it's going to move out in both directions, right? So it's not like we have DNA just being synthesized to the right or just being synthesized to the left. Rather, it's both sides of that fork at the same time. So it's moving outwards. All right, so we have that origin of replication. It's a sequence of DNA that says start here. Right? Once we start unwinding, we get that replication bubble. And on either side, where the unwinding is happening, we are calling that the replication fork. Right? Because it's forking off into two different directions. There's a lot of proteins that play a role in DNA replication, right? And we're going to talk about quite a few of them. The first one that we will talk about plays a role in this unzipping. Right? It has a role at this replication fork. That first one is called DNA helicase. Right? So DNA helicase is the protein that breaks the bonds between our nucleotides on either strand. Right? So remember, we have 3 hydrogen bonds between guanine and cytosine, 2 between adenine and thymine. It's DNA helicase that's breaking those bonds, right? Causing the separation into 2 different strands. You can kind of think of it as a zipper, right? Helicase will attach, right, at one side of the replication bubble at a replication fork. It's going to keep unwinding the DNA, right, and increasing that replication bubble. All right, so DNA helicase unwinds our DNA. Once this DNA is unwound, right, we have to have a way to keep these strands from rebinding to each other, right? We don't want it to go back into a double helix. We want to keep them apart. That's where our next set of proteins come into play. Those are the single-stranded binding proteins, often abbreviated as SSB, right? So as helicase unwinds the DNA, right, we're getting 2 separate strands. We want to prevent these strands from coming back together. And what prevents it from coming back together, what stabilizes those strands, are these single-stranded binding proteins. All right, so we want to keep those strands open because we want to use them as a template. We don't want them to rewind into a double helix. So we're stabilizing them with these single-stranded binding proteins. All right, so far we have DNA helicase unzipping the DNA double helix, and we have the single-stranded binding proteins keeping those strands in place, right? We don't want them to reform the helix. But there's one more protein playing a role in breaking apart our double helix. That last protein is DNA topoisomerase. What this protein does is it works ahead of the DNA helicase, right? And it helps relieve any additional tension that the unwinding might put onto the double helix. So I don't know if you've ever tried to like untangle a piece of string, or maybe you're pulling tendrils apart of the string, right? And the more you pull apart, the more knotted it gets, right? It just gets super coiled as you're pulling it apart. That's the same kind of concept that's happening here. And I wish I would have brought my string with so I could have demonstrated it, but I just got it, right? But if you pull it apart, right, it creates a lot of tension on the remaining helix. And so DNA topoisomerase is relieving that tension so it doesn't get knotted up or tangled up in the process. All right, so we have DNA helicase unzipping our DNA at the replication fork. The template strands, right, the unzipped strands are being stabilized by the single-stranded stabilizing proteins. And DNA topoisomerase is relieving any supercoiling, any additional tension on the molecule that's being unzipped. All right, let's take a second to review. I want you to tell me what has happened so far. What are those proteins involved?
Oh yeah, the DNA. And then DNA has. Yeah, it unwinds into it and doubles basically.
That was built for fun.
It's like
Yeah, I don't really get it. I don't know. Practice on the show
that's the second one. The first one is.
Alright, I get the sense that we're all done discussing. So, if I were to ask you, so far, what we've talked about in DNA replication, if you were to put that into one word, what has happened to DNA? Split. Yeah, what's another word we could use? Separated. I have unwound, right? We have taken a double helix and made two single strands. Right, so we have that origin of replication, right, that signifies we need to start splitting the DNA helix here. That forms the replication bubble. On either sides of that bubble, we have that replication fork where the actual unzipping is occurring. All right, what proteins are being used to do this? What one is doing the actual unzipping? Yeah, helicase, yeah. So, I thought they came up one by one. So we have DNA helicase. What about our single-stranded binding proteins? What are they doing? Keeping them separated, yeah. We don't want them to reform that double helix. And then DNA topoisomerase?
It's keeping the prevents supercoiling.
Yeah, it prevents supercoiling, right? Prevents it from being knotted up or tangled up in the original helix that is being unwound. All right, any questions so far? Let's take a look at what has happened, right? This is our replication fork, right? Our helicase is doing the unzipping. Topoisomerase is stabilizing that double helix to prevent any additional pressure on that helix. And we have our single-stranded proteins stabilizing the strands so they don't reform that helix. But there's more to it, right? Beyond just unzipping the DNA, we have to actually synthesize new strands. And that's what we're going to step into next. All right, so we have these strands that have been unzipped, right? They are being stabilized by those single-stranded stabilizing proteins. We need to start using those strands as a template. We have the protein primase, right? Primase is going to come along onto our template strands and insert a short sequence of RNA primers, which are just RNA nucleotides, right? So an RNA primer is just a sequence of nucleotides, roughly like 5 to 10 RNA nucleotides that is initially put onto the template strand by the enzyme, by the protein primase. Right? This kind of provides a starting point or a signaling point. Right? So by putting those RNA molecules there rather than DNA molecules, it signals to the other proteins that will be making the template or making the daughter strand that it needs to start making that strand at this specific part of the DNA. Now, because it's RNA, right, and we're making a DNA molecule, we're going to have to eventually go back and take out these RNA nucleotides. But for now, they signal for the start of replication. All right, so we have primase adding in our nucleotides of RNA. Right, it's just this very short sequence. We're saying this is where we need to start. Once we have that starting point established, once we have those RNA nucleotides in position, now comes the main protein, all right? This is the protein that does all the heavy lifting in DNA replication, our DNA polymerase 3, right? This is the protein that actually makes the new strands. So it's going to find the RNA primers that were added by primase. It's going to find that short sequence, and it's going to bind to that template strand. So it has to find those RNA primers, it binds to those template strands, and then it reads that template strand, right, and adds in nucleotides based on what it's reading. So remember, if it reads an adenine, it's going to add in a thymine to the daughter strand. Guanine, it's going to add in a cytosine to the daughter strand, right? So it's reading it as it goes, and as it's adding in these nucleotides, they're covalently being bonded together. Remember, what makes up the backbone of a strand of DNA is that sugar phosphate backbone, right? Those phosphodiester bonds. So we're creating those phosphodiester bonds as it reads the template strand. Now, this one has something that's not super ideal about it, right? It has something it has to overcome, and that is that it can only build a new daughter strand in one direction. It can only build a daughter strand from a 5' to 3' direction, right? And that's going to have consequences, because if we remember, we have DNA, a double helix, we've split it in half. Each of those strands that we've split run in opposite directions from each other, right? And the DNA polymerase can only read, right, can only build in that 5 to 3 direction. So what we're going to see is that we're going to have two different strands synthesized in slightly different ways. So let's focus on one of the strands. We're going to call it the leading strand, right? So we have our DNA, it's being unzipped. The leading strand has a 3' open end to it, right? Now the DNA polymerase, because it builds in the 5 to 3, right, this is ideal for it. It's just going to bind to a single primer, right, and start building that leading strand. So this arrow is actually that second strand being built, and it's just going to continue going as DNA unzips. It's going to elongate that daughter strand, right? So remember, we're starting at the 3' end. It can only build from 5 to 3, which is going to be opposite from the 3' end. Right, so keep that directionality in mind. We have 3', the new strand that's complementing it has to start at the 5' end. DNA polymerase can only build from 5' to 3', so it's just going to be a long, continuous strand being made. All right. So this is easy. This is pretty easy for the DNA polymerase. It's the other side of the strand, remember that's opposite, so now we have the 5' end sticking out. That creates problems. So we call this the lagging strand. Right? Now because we can only build from 5' to 3', we have to build the resulting daughter strand in chunks. Right? So essentially, as it unzips, we're going to come in, build a chunk. We're going to wait for more to unzip, come in, build another chunk. Right? These chunks we call Okazaki fragments. Right? It's named off of the individual that discovered this process, discovered these fragments. Right? So essentially we have fragments of a primer and a segment of DNA. And then a primer and a segment of DNA. Right? So the DNA segments are pretty short. They're only like 100 to 200 nucleotides long. And they're broken up into these chunks. All right, so synthesizing from 5' to prime, we have our leading strand can be synthesized continuously versus our lagging strand is synthesized in these fragments called Okazaki fragments. I'm going to show yet another video because I think visuals are just really helpful in understanding what's happening. So let's look at a visual of the leading and the lagging strand. Review this information, right? Answer a few questions.
So in the leading one, it just goes 5 to 3. And then in then in the other one, it starts right where it uncoils, and then it builds that way, and it like loops this, like the visual. Does that make sense?
I don't know what.
And then it like, because then it can start making up here and down here in 2 places. That's funny. That's funny. And then I don't know replicated DNA.
Is that what happens next? No, actually, it's fine. RNA primers replace DNA.
All right, we are synthesizing a new daughter strand. Which direction is it always going to be synthesized in? 5 to 3, yes. So DNA polymerase, it always reads, well, it always builds 5 to 3. It might be reading the template strand 3 to 5, but it's always building in the 3 or the 5 to 3 prime direction, right? Always from 5 to 3. All right. How is replication different in the leading versus the lagging strand?
Lagging strands have Okazaki fragments.
Yeah, so lagging strand has the Okazaki fragments. What about the leading strand? It's nice and continuous. All right, so we have fragments. We have a nice continuous strand. We have some RNA primers in there. What do you think has to happen next?
The RNA nucleotides will be replaced by some DNA.
Yeah, we have to replace those RNA nucleotides, right? We're building a DNA molecule. We need to only have DNA nucleotides. We have to replace those primers that were placed by the primates. I forgot I had that up there. All right, I'll leave it on here for a couple seconds in case you want to write it down. All right, so again, the legging only needs one RNA primer because it's a nice continuous strand. All right, once we have the primer added, we're going to put on the polymerase, synthesize a nice continuous strand. Versus those Okazaki fragments that are being made, we need to have multiple primers because we have to make multiple strands, multiple fragments. All right, so Primase has added on those RNA primers. We have to get rid of those RNA nucleotides. We have another polymerase doing the work here. So this time it's DNA polymerase 1. Right? Remember, 3 is the one that builds. 1 is the one that goes in, takes away the RNA nucleotides, adds in the appropriate DNA nucleotides. Right, so it's taking out the RNA primer, replacing it with the DNA molecules, DNA nucleotides. The one thing it can't do is join the strand of DNA nucleotides it is just added to the strand of nucleotides, DNA nucleotides, that were synthesized by polymerase 3. Remember, we had a primer, we had a segment of DNA. We had a primer, a segment of DNA. We can replace that primer with DNA molecules, but we can't actually combine that newly synthesized segment of DNA with the one that was previously segmented. Right? So we can't attach them together. So again, if we're looking at our diagram here, we have topoisomerase stabilizing the double helix at the replication fork. Helicase is doing the unwinding. SSB proteins are stopping them from reforming the double helix, stabilizing the strands. Primase is adding in the primers, which then signals for DNA polymerase 3 to come in and start synthesizing a new fragment, right? The lagging strand, we have to synthesize it in segments. Leading is continuous. DNA polymerase 1 is going to come in, remove those primers that were added by the primase, and make them DNA nucleotides. There's one more thing that we have to do. We have to put it all together. All right, so ligase is the last protein here. Ligase you can kind of think of as like the glue that will glue all of the fragments together. So now we have one continuous line in both directions on both the leading and lagging strand, right? It's gluing those fragments together, creating those covalent bonds in the sugar phosphate backbone. Right, so ligase is putting it all together. All right, so we've gone through all the steps of DNA replication now, but I want us to watch a video that just puts it all together. I will say this video is a little older. You'll notice that when it talks about the polymerase, it doesn't say polymerase 3 versus polymerase 1. It just says the polymerases, all right? Let's go ahead and all right, just a couple more slides here, and we're gonna end on time. So this replication process is very accurate, all right? And there's 3 different ways or 3 different reasons behind this accuracy, right? One is just simply the hydrogen bonding, right? Hydrogen bonding between A and T, and then between G and C, right? It's just when they're matched appropriately, those hydrogen bonds are more stable, right? So if you match an A and a C together, those hydrogen bonds are less stable. Right? And we like stability in our molecules, so they're always going to want to be stable. Right? DNA polymerase, right, is really good at proofreading, right? And it's very unlikely to form bonds if a pair is mismatched. Right? So it kind of proofreads itself as it goes along and adds new DNA nucleotides. And then DNA polymerase can also, especially polymerase 1, can go back in and proofread what has been added. So even though DNA polymerase 3 is really good at what it does, right, and very unlikely to form a mismatched pair, DNA polymerase 1 will go back double check the work, proofread it, and if there is a mistake, it has the ability to remove mismatched pairs and replace it with the correct one. Right? Now there's a few other methods as well that helps in proofreading, but these are some of the 3 main ones. All right, I want to end with doing this slide, all right? So take 3 more minutes here. What are the proteins used in directly building the new DNA strand, right? Not just unzipping it, but directly building that new daughter strand. Oh, the timers are not working out very well today. Roughly 3 minutes. Alright, I feel like it's been roughly like 2-ish minutes. So let's answer this slide quickly here. What proteins are used in directly building a new strand? What's one of them? Primase. Another one. Polymerase 1. Polymerase 3. Ligase. Yes. So they're all used in directly building that new strand. All right. Homework for Friday. I do have a content check out there. It should be something that could be done before Friday, definitely. We'll do the rest of it in class, and it will be a drawing in class assignment. So keep that in mind if you want to print it or do it digitally. And then exam corrections, again, due Monday.